Can everyone hear me? I think we'll get started. Actually, Jonathan, can you dim the lights, or at least on the front part? Just I know Jennifer's gonna have some nice, pretty images, and we make sure we can see them. So thank you all for coming. I think there might be more people coming. I have some students that are for my class that I think are coming, and they're probably trekking their way across campus. But we want to get started, so we get on time and we let Jennifer eat lunch after this. So thank all of you for coming today, and thank you very much. I'm really pleased to introduce our speaker today, Dr. Jennifer Sun. She's our next candidate for our School of Neuroscience faculty recruitment. So Jennifer is coming to us from California. She spent the last several years in California. She did her PhD at the University of Southern California, and then currently is at University of California, San Francisco. And throughout her career, she's been focused a lot, she'll be telling you about a bit today about her career path. She's focused a lot on looking at sensory systems, particularly visual systems and auditory systems, looking at how these circuits are built during development, doing both systems neuroscience and some computational modeling, understand how these systems come together, but also going down to the molecular level to understand at a synaptic plasticity, molecular cellular level, what are the changes that happen as these circuits are being built in the developing brain and how they function. So she has amazing credentials. She has many wonderful papers. Pretty much, I think every single one of her papers is in one of our highest impact journals in neuroscience. So I was a little bit jealous of the CV when I was looking at it. Sorry, Jennifer. So a really amazing publication record that's really been building over time, over the course of her career, even from undergraduate research up till now. She's been building a really nice story in this series of tools and expertise that she'll be telling you about today. Her work has been recognized too. She has a very prestigious K99 R00. It's one of these grants that can support both your postdoctoral work as well as your it's from the NIH and support your transition into building your own laboratory. So that she is awarded one of these, and that would help to propagate her into moving into an independent research career. So in addition to grants and publications, she's also spent a great deal of time working with students, both mentoring them and working with them in the laboratory, teaching them in the classroom. So I feel like in many ways she would fit in really nicely with the group, the faculty that we have here already at Virginia Tech. So without further ado, I'll let her tell us about her work. Thanks.
Thank you. Thank you very much for the kind introduction. And also I would like take this opportunity to thank the interview panel for inviting me here. It's really exciting to be at this beautiful campus and also interacting with all the aspiring young scientists and students in this very energetic environment here. So today I'm gonna spend some time talk about my past research focusing on the development and plasticity in the auditory and visual systems, the two most indispensable sensory modalities that we rely on to navigate through our daily life. Thanks to our highly competent visual system, we can distinguish a flying football from human body parts and track its motion in a split second. And our hearing enables us to listen to one another and engage in a conversation. And both auditory and visual processing are so essential that almost it's required for all the complex and the cognitive functions in our in our brain that would enable us to process emotion, decision making, and also learning and memory. So, and we know that the impaired processing would severely affect the life quality. And it's commonly seen in patients suffering from various diseases in both young and adult. For example, according to the NIH, at least 2 to 3% of the children would develop amblyopia, also called lazy eye, which is actually the lazy brain that fails to process the visual information from retina. And also, it's actually very common in the stroke patient that they suffer or experience certain type of vision loss. And at least 1/5 of them would end up with some deficit in the visual processing. Therefore, in the past decade, my research has centered around this goal to really understand the neuron basis of development and the plasticity in the sensory cortical circuits. So in particular, I asked how the auditory and visual cortical circuits are wired during development to achieve their highly functional and specialized properties. Also, I want to ask how miswiring and rewiring happen in response to altered environment in the developing as well as mature brain. So in the process, I hope to uncover the general mechanism from the circuit to molecular level, and ultimately that can be applied to help patients with those sensory processing disorders. So first, let's take a look at the brain circuit for sensory processing. Those visual and auditory cues from the external environment are first converted to electric pulses by those sensory receptors in the retina or cochlea, which are then relayed directly or indirectly onto those thalamus, and finally pass to the primary and higher sensory cortices, where the perception actually happens. And if we zoom into the sensory cortices, we can see there is a stereotypical 6-layer structure that's almost present in all mammalian species. And the neurons in the layer 4, also known as the recipient or input layer, would receive the majority of the thalamic input. And then they pass it on to upper layer 2-3 and further down to the deep layers, both of which would send their projections out to other cortical areas and subcortical areas for processing. So this laminal structure really grants each layer very distinct properties for sensory processing and development plasticity. So in the next 20 minutes or so, I'm going to present part of my research in developing layer 4 auditory cortices and also the layer 2-3 visual cortices. And then I will talk about my current and future program, study the adult visual plasticity regulated by diverse types of neurons and molecules in the local microenvironment, as well as distributed brain circuits. So first, I'll talk about the refinement of sound representation in the auditory cortex, which is a characteristic development process that's happening in almost all the sensory modalities. In fact, in the autistic children, this process is somehow disrupted so that they have hypersensory processing, which is at least a symptom and possibly the cause of many of the behavior signs that's shown in those autism spectral disorders. So in order to really look at the neurons' responses to the auditory responses, people have been using this technology invented by the Nobel laureate Hodgkin and Huxley back in 50s. Those using a pipette to detect the electric discharge from a neuron, which can detect the changes at the level of millivolts. So used this way, I can stick the pipette into the auditory cortex and then present different tones to the animal while recording their neuronal responses to that specific stimulus. And then after a repetitive measurement of all different combination of tones at different frequency and intensity, and then arrange them in a map where you can see that there's the sound frequency and intensity level at the X and Y axis, I can map up the neuron response using this method. So what you can appreciate in this reset field or the responding sound map is this particular neuron, when the sound gets louder and louder, it responds to a wider range of sound frequencies. And at the lowest sound responding frequency, we can look at the range of the frequency it responds to. In those typical adult neurons, they would respond to a sound range of about 1 octave, which is like in music from middle C to high C. And but if we really look at neurons in younger animals, they experience much wider response. So especially you can appreciate in this case from the postnatal day 16 to 22, which is just less than 1 week, they have this dramatic refinement of their sound representation. And my research has been shown that this process happens probably at the cortical level, because when we recorded the output from the thalamus, you can see that this sound map at each individual neuron didn't change. So that we just focus on the cortical level to see how the synaptic interaction would give rise to this property during development. And if we look at the layer 4 of the auditory cortex, not only they receive the direct input from thalamus, there are also local interneurons and excitatory neurons that would send their projections to the pyramidal neurons that we are recording, so that there's the synaptic input of the excitatory neurons and inhibitory neurons. And instead of doing the loose patch, we can do this whole cell recording by putting the pipette in there and clamp it at different voltage, so that we can separate those input contributed by the excitatory neurons or inhibitory neurons. And by doing so, I can get this synaptic map of the individual neuron. So I can look what it looks like for the input from those excitatory sources versus the inhibitory sources. And in my past research, I have demonstrated that during this refinement, there is a dramatic sharpening of the excitatory input, and the inhibition actually stay more or less broadly tuned, so that after integration, which I use a computation model to combine and calculate their spike responses and generate their output responses, you can see here that it really recapitulates this significant functional refinement that we observed in the animals. And if you noticed earlier that when I show you the excitation, it actually comes from 2 different sources. Mainly all the thalamic input are excitatory. And also there are a lot of recurrent excitation happening within the layer 4. So in order to separate these 2 different resources, see how they interact or contribute to this total refinement, I applied the drug muscimol to temporarily silence the cortical responses, so that afterwards I get this sole contribution from thalamocortical input. And in this way, by comparing the difference of the 2 within the same neuron, I would also be able to generate the intracortical contribution that is shown by this pharmacological manipulation. And if we look at one neuron that at this level, for both its thalamic input as well as its intracortical input, we see that there is a dramatic decrease over time for both sources. And if we really look at further into their amplitude ratio of the 2, we notice that at early stage, the intracortical input is much weaker than the thalamocortical contribution, versus in the later stage, the intracortical recurrent excitation become much, much more stronger and dominant over time. Therefore, in my study, I've shown that the intracortical excitatory input are selectively narrowed and much strengthened over this development, and leading to this much selective and refined tuning for the excitation. Meanwhile, this inhibition stay relatively more broad, and after the integration of the 2 different resources, they would be able to further produce this much sharpened and refined sensory responses. So what I've shown so far is what varying process of the sensory cortical circuit during normal development. But what if there's abnormal experience during some environmental or internal conditions that prevent this process? What would be the circuit wiring and rewiring being damaged or perturbed, and is it possible to reverse this process? To understand these questions, I've been using the visual cortical area to study the ocular dominance plasticity, one of the most successful and impactful model ever used since its first description by Hubel and Wissel. And that's due to several reasons. First, we know that the primary visual cortex is the very first station where the binocular vision, input from both eyes, converge at the level of single neuron. Therefore, it allows us to really isolate the cortical changes from those taking place at the earlier sensory processing stages. Moreover, in Hubel and Wissel's phenomenal work, they demonstrated the monocular deprivation by this eyelid suture would really cause drastic change of ocular dominance towards the open eye during early development, which is a great case and very similar to the human amblyopia condition, so that it's a great framework to study its biological underpinning for the clinical condition. And over the years, many scientists confirmed that this is a very well-preserved visual developmental process in many different species, varying only in time specific to their maturation speed. So it applies really to from monkey to cat and ferret, even mice and rats most recent time. And also the mice, which we all appreciate that they offer us such a unique opportunity to really dissect down into their cellular and molecular properties because of the genetic tools that's becoming available nowadays. So we're really at a prime time to use these tools and use a mouse model system to study all its underlying biological processes. So in accompanying of those physiological changes, has been shown that there's dramatic anatomical shift within this first 4 or 5 days, and that's beautiful data in the cat visual cortex, where they see this massive shrink of the thalamocortical arbors projecting from specifically the deprived eye, and that would result in a huge loss of at least half of the input. And interestingly, it's later discovered the physiological changes beyond the input layer, or even faster, taking only 2 days in cats layer 2-3, and up to 3 days in mice. So you can appreciate this. Within the layer 2-3, there's a dramatic shift of the overall population towards the ipsilateral eye, which is left open. And by then, there's not much change at the layer 4 yet. So we know that it leads to an immediate question. What would be the basis for this fast shift at the upper layers without the layer 4, which is the major feeding layers to layer 2-3, have any changes showing up? So to answer this question, I tagged the green and red fluorescent protein to the postsynaptic spines, and in those sparsely labeled layer 2-3 pyramidal neurons to visualize its excitatory synapses at each single dendrite. So now you can appreciate this little lit puncta structure, which is the protrusion of the synapses between the excitatory neurons that are all nicely labeled for the sparsely labeled neuron, so that we would be able to track those neuronal changes over time and look at how each neuron would evolve and accommodate and enable the changes that we detected in its functional responses. And I did this by doing the typical ocular dominance monocular deprivation in the animal for 6 days of visual depression by eyelid suture in the contralateral eye, and give it for 2 days to recover to see how at each time point the neuron would change their structure to accommodate their functional responses. And also, so there are the same neuron along the 8 days of time course that I would be able to acquire those, track these spine changes over time. And then I use a set of pipelines set up and developed through my lab members and me to look at how these neurons would be changed over time. We overlay the neurons, the same neuron over days, and then doing rigid translation as well as warping to accommodate the changing brain at this very young stage. Notice that it's still developing, so there's some warping and deformation of the brain happening. And after that, I developed this Fiji plug-in system where it can automatically track the spines and compare their formation or the elimination over different days. And what we found that apparently there is a dramatic shift or decrease of the spine densities in the animals that going through this monocular deprivation. And interestingly, it comes back to the normal level and stayed more or less the same during recovery stage. And that it's very consistent to what people have been shown in the population data, where they see that the response coming from the contralateral eye, the sutured eye, significantly dropped after 3 days. And then there's an overall compensatory process happening if the ocular, the deprivation persists longer. And then after this recover, it comes back. And this process doesn't really happen in the animals that just going through the normal development. Their spine density would stay more or less the same. And to really further look into the individual neurons and study how their structure and function would correlate and whether it would provide us direct evidence for its response changes. We actually track the neuron response change, and then I do this behavior physiological mapping where I present the visual stimulus to each specific eye in this anesthetized animal, and then recorded their neuron responses by removing the cover glass in the brain after I finish all this structural imaging. So in this way, we can calculate its neuron responses and its preference to the input from each specific eye and calculate how this neuron prefers the input from contralateral or ipsilateral eye. We can see that for this neuron that have some dramatic loss of the spines only after 3 days of monocular deprivation, they have a good preference towards the contralateral eye, which is being sutured. And that explains how this synaptic density decrease because of the loss of the overall input drive. And indeed, after we measure 9 neurons that experience certain different degrees of spine changes, we see there is a very nice tight correlation of their spine structural changes versus their preference to the eye from the ipsi or contralateral eye. Therefore, I've been showing that this spine remodeling corresponds with the plasticity changes that we observed at the physiological level in both the population response as well as the single cell level. And in fact, this spine remodeling, it's not a really brand new idea. It has been previously extensively studied in the adult brain. One thing, it's really easy to track the adult brain because they don't change anyway. You can really look at the single neuron. But it's also because people trying to know what is going on in the adult brain. Is there any possibility for them to make plastic changes at all? Because it's funny, there's a comment made by San Diego Romani Cajal ages ago, and he's really have this pessimistic view that they're saying that in the adult brain, the nerve passes as something fixed and immutable. And it's really up to the future generation to figure out a way to help it evolve and change. And indeed, we know that that's not completely true, and but even the changes in the adult brain is much slower and also smaller in terms of its amplitude. That would account for like very difficult recovery in patients who suffer from the stroke, or the patient who have the amblyopia in early age and left until they are in adult. It's difficult, but we know that there are a lot of ways going on and a lot of research that have been developed over the years to trying to apply all different pharmacological or stem cell transplant and electric stimulation. All sorts of efforts have been investigated to see how it can help the adult brain to become a little bit more plastic. And in a recent study by the Stryker lab, which is my current lab at UCSF, it's demonstrated that there is a simple way that could increase the adult cortical plasticity even in the adult mice, which is running. That's something a little bit unexpected and also very comforting to know. And but you do need to know that this running and the visual exposure need to be sort of in the same time range of the 4 or 5 hours, because what they did for a control experiment, that they let the animal run for a few hours and then put them back in the cage and let them just staring at the screen where the visual stimulus had been delivered. There's no significant enhancement anymore. So that really indicating that there is a time window that's restricting this running effect on the visual plasticity. So to determine if there's a concurrent running and visual experience is really required, I designed this closed loop visual training paradigm to deliver the visual stimulus to animals depending on its locomotion state. So you can see that when the animal is running, there's a drifting grading of certain orientation is being presented. And when it's still, there's a different type of visual stimulation is being presented. So, and in between, there's a gray screen just facing out to make sure they receive equal amount of stimulation over the whole time course when the animal is on the ball. And after that, for 5 days, about 1 hour training every single day, I look at how the neuron responses change over time, that I present 8 different orientations of visual stimulus to the mice before and after this 5 days training paradigm. And then I can record it like 500 to 600 of neurons in one animal at different planes that are within the layer 2-3 visual cortex. And then use this pipeline processing to really identify each individual neuron and also compare their changes within each individual neuron. And what you can see here that I have those neurons tracked before the exposure and after the 5 days of exposure, and then I pair their responses that in response to different visual stimulus orientation. That's indicated by those colors. That's really a short amount of time for like 20 minutes or so. I can give 15 repetitions of visual stimulus to get a very reliable visual responses where you can see after those training, the animals, the same neuron that have the visual preference at the beginning would have much clearer visual representation at each orientation, particularly if we look at those orientations that's being presented during the training, where the red label ones are the ones that being presented exclusively during the animal is running, versus the blue ones are the ones that presented when the animal is being still. You can see here, there's very dramatic increase for the visual responses to those running stimulus in this neuron, versus for the still conditions, they don't really have significant enhancement. And over hundreds of neurons, indeed, there's very significant enhancement specifically for this presentation being presented during running states. So what I've been showing that is really this running, it's a critical and helpful tool to help the visual neurons could adjust and compensate those rigid environment in the adult brain and try to enhance their responses when it's needed or it's right timing. However, it's still not really clear how the running would exert its direct effect on this temporally tied regulation. And there are several hypotheses. One of them, actually several of them, will rely on this neuromodulatory pathways, because people have been shown that when the brain is being aroused, there are a lot of centers that directly representing this enhanced arousal state, or even the running, which is the extreme case for arousal, would engage all those deep nuclei where they send their neuromodulatory input all the way to the neocortexes, including all the visual cortexes, where the response can be modulated. And that likely would contribute to their long-term modification of their responses over time. And in my future research, I would really like to dissect each individual pathways, for example, to look at the dorsal raphe by expressing varus, and then look at how their responses would correlate the behavior state and how their projection up onto visual cortex would be able to specifically modulate the visual cortical processing there and eventually lead to the visual plasticity in these adult brain. And one more question I want to study is to use the dual cell patching, which is expertise I drawn in Roger Nico lab, where I can look at how the excitatory or inhibitory connectivity would be affected by activating those specific fibers coming from the neuromodulatory pathway, and see that beyond the fast neurotransmitter, how those relatively more diffusive and long-lasting neuromodulator would contribute to this process. And another project, or the aspect of this visual plasticity in the adult brain, is to look at their local microenvironment happening within the visual cortex. As I mentioned earlier already, that there's the excitatory neurons, and also there are the inhibitory population, which have been shown nowadays is very diverse in terms of their type and their anatomical properties. And before, it's really hard to look at them because they are very sparse, consisting less than 20%. And also, they're relatively smaller and harder to really approach directly with the electrode. But nowadays, with all the transgenic lines in mouse, we can really distinguish each individual cell types and use the Cre lines to really nicely identify each one of them, and together use these markers and the calcium indicator that's expressed in the animals. We can track their changes together with all other excitatory neurons that's nearby and see how their interaction over time will lead to this functional enhancement in the adult neuron. And since I'm talking about the interneuron, I would want to bring up this topic where I get involved into a research study of the glia cells, where I would be able to help identify some physiological changes at the neurons and glia cells, which actually share the same precursor cells in the brain. And interestingly, those glia cells were actually have very tight interaction with other interneurons, and they are a great spot to store all the neurotransmitters within themselves, which would potentially be a great and important player in regulating neuron plasticity. I'm sure there are many in-house glia biologists know much better than me to tell you how important they are in both health and disease conditions. So that's my main research theme. And besides that, ultimately, I want to understand how it works in the normal developing and adult brains. And ultimately, I want to bring this understanding and technique to form a collaboration with the clinicians and bioengineering to really apply it to help people recover from the state conditions, disease conditions. So one of the research I've been collaborating with bioengineering is we look at how the brain stimulation would effectively recruit neurons of both excitatory and inhibitory population, and then maybe administrate different effect on those population. And by understanding their selective effects, we would able to deliver some more efficient design and deliver some more efficient stimulation patterns that can better recruit or facilitate the rehabilitation in patients. And another area that I'm really interested in is to look at the stroke condition, how the vasculature would remodel over time, and how they would, again, interact and affect those glia cells and the local interneuron and excitatory neurons to eventually bring up all this technique together and then apply it to some translational research. So just a brief summary that we're really at this stage that everything have come to a prime time for us to look at the brain not as a single isolated spot, but it's really an interconnected and a super dynamic system where the visual cortex not only receive the input from the retina, but also they send the feedback response and they crosstalk with each other in all the brain areas. And also there's this neuromodulatory centers from the deep nuclei that would project all through the neocortex and affect those sensory processings in all the cortical area. And more importantly, at this microenvironment, there are way more factors playing in other than the excitatory neuron. That's the only accessible factor before. Nowadays, we know that there might be way more behind of the scenes players that we would want to know how we could explore their functions and the strategies to develop and design better treatments. So with that, I would like to really look into the future that we can better understanding those underlying biological process and find a good balance or a way for the brain to regulate its stability for the reliable sensory representation, as well as when we need it to be a little bit more flexible and plastic so that we can enable it for efficient perception learning and also the rehabilitation. With that, I would like to thank all your attention, and I'm happy to take questions. Yes.
I think your result was really nice. When I think about neuromodulatory responses and locomotion,
Yeah.
I think about the locus coeruleus and neurogenesis.
Yes.
Why did you choose the rat nucleus for your example to study first?
So it's a great question. Yes, locus coeruleus have been shown very effective in driving the running activity and recruiting. So one of the reasons I choose, I'm definitely gonna explore all these 3 different neuromodulators that I presented here. But one of the reasons I'm looking at the serotonergic input is because during early development, there's massive projection from the dorsal raphe directly to the input layers that presumably might play a big factor in regulating and putting the developing brain in a much more plastic state. Versus in the adult animals, some early anatomical studies have been shown there's a drastic decrease of those input at the input layer. But there are still some reasonable number of fiber ABERs projecting to the upper layer. So I think that will be a really good starting point to dissect the upper layers and see whether that's one of the major reasons that they are still maintaining their plasticity.
With regards to the coincidence of the running with the visual stimuli and the learning or refinement plasticity that takes place, would that suggest that if you wanted to enhance visual learning that you'd have to stand on the treadmill while you're working on your computer, or that it's really only what you're perceiving at the moment that you're in the active mode of exercising that you get the benefit from it?
That's a really good point. Actually, there are some clinical trials and papers publishing saying they have the patient not like running on a treadmill, but doing a static cycling. And then they deliver the visual input to the specific eye to try to help. And seems like it's working, but it's a little bit controversial at the moment. There's some group in Italy that's showing that's very effective, and the other group in UK saying like that's the way their assessment is not yet good enough to convince other people. But yes, that's definitely one of the indications from our research that there seems to be a tight correlation that's required to achieve this plasticity. But I think what I hope is to, by understanding its underlying mechanism, potentially through different pathways, that I can eventually directly recruit those neuromodulatory pathway. So for patients who cannot run, there would be an alternative way to stimulate them and then enhance their visual responses. Yeah.
Along the same lines, so I just want to make sure I'm correct on this. They run first for the control. They run first and then they're given the visual.
Yes. Yeah.
We tried it the reverse because I was thinking in associative learning you have to have the cue before. Otherwise, what if it's not just the running, but it's the association between the running and the cue, right? That's increasing the ability to learn the signal for the cell. So if the cue's first and then you, like if there's something dangerous and you run away from it, right? You learn that thing's dangerous.
Right.
So instead what you're giving them is the running first and then the cue, so there would be no association. It would be like specifically unpairing them. So have you tried that as a control?
I see. So you're suggesting
They have to have the visual first and then the running. Right?
it's a really, it's a very interesting and reasonable point because usually people do the reward or the learning training. They give the animal some task and then they deliver the reward.
Right. But if you're doing like idling conditioning, right?
Yeah.
You have the sound and then if you have the shock first and then the sound, they'll never learn it. In fact, they learn the opposite relationship.
Right. So it's a little bit complicated here in terms that running it's a natural behavior. So, but that being said, there might be a way to deliver the running as a forced environment that the treadmill would started rolling afterwards. And yeah, that's something that we didn't incorporate into our recent study because the animals sort of have this at the wheel running at the moment, and we want to just deliver the stimulus to see how the neuromodulators would potentially bring some regulation in a natural setting. But if we were to do this forced one, I think I should also look at into the dopamine center and other reward area that see how they project into that. Yeah.
Flipping it backwards like that almost makes it more like instead you're looking at two different processes then, right? Like you're almost looking at what is the effect. Think of it not in terms of conditioning, but in terms of just the neuromodulatory effect. You're almost looking at what the difference is between like applying neuromodulators while the activity is going on versus the other way is having a behavior and then like during the consolidation or the later biochemical processes, the modulators. So yeah, I mean it would be interesting to look at it both ways. I think because
So
tell you about potentially the locus of the effect of the neuromodulators.
So I talked to some reward or behavior training scientists, and one thing they did mention is even for reward, it's better to like do it right after, so that there's apparently a time window to give the reward for the animal to really associate to different things and knowing there's the consequence of the prior information. So there's also like a lot of bioengineering study that instead of doing running or this, they do a very similar but much more invasive procedure to do the vagal nerve stimulation, which is for sure recruiting all those modulatory centers. And what they found is also there needs to be a very tight temporal correlation between the cognitive training as well as the stimulation. So as if they're almost at the same time. So I think what we are delivering here is a visual stimulus that's definitely exclusive in each behavioral state, but it doesn't really decorrelate in terms that one comes first and another. Most cases for my visual stimulation, I give a time window to make sure the animal has been running a little bit, and then I deliver the visual stimulation for a short period of time, but usually I think that's the animal is still running a little bit before it comes off.
Right.
Yeah.
If you were running away from it, right? Let's say there's a bear and you run away from it, that you're going to associate the bear with why you ran, as opposed to if I'm running and then I see a bear,
Right.
right? It's going to be a very different experience. You're not necessarily going to make that association that it's run away.
It's interesting, yeah. And I think reward learning and fear learning, it's also a little bit different in terms, there's some like spine decrease that usually associated with fear learning versus for the reward learning, it's normally some like an enhancement or so. But yes, it would be a very interesting and challenging way to design this experiment to explore that. But I think Matt have been, yeah, you have it.
Just want to first say, this is a really nice talk. I really enjoy seeing this talk.
Oh, thank you so much.
My question is, a lot of the systems that you're talking about with your wheel running, you're suggesting involve monoaminergic systems. Do you think that you could recapitulate this pharmacologically by putting in, let's say, an SSRI or cocaine and trying to pair that?
Yeah, so the short answer, yes, there are some people that have been doing similar things where they give the serotonin uptake inhibitor, and then they can see that the animal the adult animal definitely become a little bit more plastic and susceptible to environmental changes. So the short answer is yes, there would be some available drugs that we can directly test onto it. But also, I personally would be interested to look at how the circuit level, like the crosstalk between each brain areas to do. But eventually, they have to come down to this microenvironment, how the neurons and their receptors there would take into those drugs and then manifest their responses eventually in the excitatory neurons.
So if I need this in the future, I don't have to run, I can just take cocaine.
Yes, I know, yeah.
Yeah, so I think the change in the neuromodulators and plasticity is interesting, especially since they're also regulated during the sleep cycle.
Yeah.
Another line is that you were talking about two things that you talked about different areas, and you talked about coupling. So you're probably familiar with, Charlie Schroeder and Peter Lakatos's work where they show that you can get cross-frequency coupling, right, across different modalities, right?
Definitely.
So visual stimulus can re-entrain the delta and the higher frequency oscillations.
Yeah.
So since you're doing locomotion and visual, have you thought about, okay, we're looking at this chemical level, but how important is there some sort of oscillatory coupling between, a motor area and the visual area to drive the plasticity, right? That seems like that could be a really other line of research where you're looking at even a more global level that could be very critical to the plasticity.
That's a really great insight. So actually, there is sort of suggestion at the thalamic level. Their firing phase would change a little bit. That potentially would also increase their efficiency to talk to the cortical area, and that make them a little bit more effective in changing the responses at the cortical level. But that would require some due recording at both sites. And I think that would at least give us a really great starting point to look at how the excitatory neurons crosstalk with each other. And then we can assess it with the local environment for its interaction with different neuron types.
That
Yes.
would have some nice translational applications, right? Like for autism and stuff like that. You could probably
Stick electrode into the brain. Yeah.
No, the cross-modal conversation between the areas and how the coupling might relate to plasticity, right?
Yeah, yeah. So yeah, that's also a great aspect that for the autistic kids and how to engage them a little bit to use more brain area or so. Yeah. Yes.
Actually, that's perfect,
Mike.
I was going to ask something similar.
Yeah.
So a lot of these neuromodulatory inputs also go to thalamus.
Right.
And could kind of alter, we now know that there's oculodominant plasticity in thalamus, kind of new, and that could then alter cortical oculodominant plasticity. So how do you see being able to tease all this apart with the tools that you're generating to study the neuromodulatory circuits?
Yeah. So first of all, I definitely agree. I think the neuromodulation have some direct effect on the thalamus, and I think one area I definitely want to go in the future is to directly record the thalamic neuron, the LGNs, and see how they're changing their responses. I've been doing a little bit in the lab, and I do notice with the running, their responses is switching a little bit from the phasic to more or less like tonic, and that would definitely shift their frequency and their synchronization with brain centers a little bit far away from it. And I think in the future, this line would directly nicely go from this viral injection at the cortical and deep neuron layer to the cortical to the deep neuron to the thalamocortical projection. And with that, I would be able to just, again, use the optogenetic and see just how these neurons being affected, and also whether there's a developmental shift over time, because we know that those neuromodulatory pathways tend to have a very temporal sensitive regulation over the development, and that's one of the things that I want to explore in the future. Yes.
Perfectly on
Thank you so much. Yeah.